two talks on the biology of Dictyostelium or slime mold. You remember that we had this modeling talk yesterday, the last talk of yesterday, where Trilochan showed, a model of how Dicty does cyclic AMP signaling, and there's a streaming motion, and then this slug forms and so on. So, but he didn't go into the biology at all, and that's because Baskar and later on Professor Nanjindia is going to give details of this. So it's a pleasure to introduce Professor R. Baskar from the Department of Biotechnology, IIT Madras, which is basically our neighboring institution. So Baskar did his PhD with Professor Vidyanand Nanjindia at the University of Science, Bangalore, and then he went for his postdoc at Cold Spring Harbor Lab, where he worked with Professor Willy Gross Nicholas, who's also visited IMSC actually a couple of times, right? Thanks to Baskar. And then after a short stint at NCBS, Baskar joined IIT Madras in 2004, where he runs this plant lab, which looks at very exciting things. And it's been a pleasure to, discuss several times with Baskar on his very interesting projects. And today Baskar is going to give a talk which is partly going to be introduction to the Dictyostelium biology and partly going to talk about his own research. So without further ado, let me invite Professor Baskar for his talk.
Thank you. Thank you so much, Kabra. So we work with two fantastic model organisms. One is Dictyostelium. This is a social amoeba, and also a model plant called Arabidopsis. And Polyspondylum is the model that I started but later gave up because of various constraints, because this was not easy for us to transform. And I'm afraid that this talk is going to be completely out and out biology. If you don't understand at any point of time, please don't hesitate to stop me. So I'm sure you must know, some of you may not know, or some of you may know what is embryonic organizers. This is a small subset of region within a developing embryo, a vertebrate embryo. The red region is highlighted here. If you transplant this portion of this, the red portion, and then transplant it to a chick embryo, another embryo, so finally you get two head formation, which is there in a different model here, chick model, as well as a model, it's an amphibian model. So this region, the red region that is highlighted here, this is called an organizer. The organizer is a region of a subset of cells within a developing embryo, vertebrate embryo, which induces the neighboring cells to take on a different cell fate, and it gives you a polarity. It establishes the polarity. Without an organizer, the organism will not be developing further. So if you have a block in organizer development, completely the development will be blocked. And the second term that I want to introduce is about morphogens. This morning I heard someone talking about morphogens. I'm sure you must be knowing what is a morphogen. So these are gradients of substances along the length of the embryo, which is established and stays as it is. So now you can ask me a simple question: can the gradient be, because of diffusion, can this be diluted out? But that doesn't happen. There are various mechanisms to restrict this gradient, and the concentration gradient of these embryos, concentration gradient of these morphogens along the axis of the embryo, developing embryo, determines the cell fate. For example, the cell fate, this fate that is the fate X, Y, and Z, that depends on the concentration gradient of these morphogens along the axis of the embryo. So now comes Dictyostelium. I want to run a short movie here. Some of you may have seen this. This is a motile stage. This is a multicellular stage of Dictyostelium, which is mobile on a agar substratum. This is called the slug. This movie is from John Bonner, who was a pioneer in Dictyostelium research. This is a single-celled amoeba, which is grabbing its food and then looking for food. So now they are running out of food. If they run out of food, something interesting happens. All the amoeba converge at common collecting points, and then because of cyclic AMP chemotaxis, because of signal relay, one amoeba attracts each other and then forms a structure called an aggregate. So this aggregate of cells slowly turns into a motile structure called the slug. So now the aggregate becomes what is called as a mound. The mound, then it undergoes further morphogenesis, and then it becomes what is called as a fruiting body. So the 20% of the cells in the slug, the anterior 20% of the cells, roughly the size, roughly the anterior 20% of the cells, they go through programmed cell death and forms a dead stalk, while the rest 80% are lifted in the air and forms the spore mass. So this is another movie, which is a very nice movie I want to show. So here you add up, this is the bacterial plate. Here you add a drop of Dictyostelium cells. So they eat and it forms a fruiting body. Those are the next round of cells, it forms like this. This is very cinematographic or whatever. So you have a colony of Dictyostelium. If you initially spread a lawn of bacteria, eventually all the lawn of bacteria will be eaten away, and then the slime molds, forest of slime molds will be formed. You get fruiting bodies here. Here you have, so what you have is a circular plate. In the center, you drop a piece of Dictyostelium cells. What happens is they keep eating. If they keep eating, then the circumference also goes up.
Yeah, so that's that visible wavefront that we are seeing. Is it like the depletion of the bacteria?
It is absolutely depletion of bacteria.
Oh.
So to summarize the life cycle, so as long as there is food source, they keep eating and proliferating. So once if the food source runs out, something interesting happens. So the amoeba becomes aggregation competent. So the cells converge at common collecting points. It forms what is called as a mound. This mound slowly forms a structure called as a tip mound. The tip here is functionally equivalent to the embryonic organizers of higher vertebrates. Then this tipped aggregate slowly turns into a structure called a slug. The anterior 20% of the slug, they die and forms a dead stalk here, and the rest 80%, these are called pre-spore cells. They form dormant spore mass, spores. So the embryonic organizers, the functionally equivalent organizers of higher vertebrates, was discovered in the year 1975. And if you transplant the tip of Dictyostelium slug to another slug, you eventually get two-headed slugs, two slugs from the single body. So at various sites, this transplantation was done, and they found in all cases there are two slugs emerging from a single slug. So this was identified, even though organizers were largely confined or originally discovered in vertebrates. This was also functionally equivalent organizers were also identified in Dictyostelium, and the various morphogens along the slug axis were the well-known morphogens include cyclic AMP, adenosine, ammonia, and DEF. Cyclic AMP is a chemoattractant. It is also a first messenger. Everyone knows it's a second messenger, but here it is acting as a first messenger. Then the breakdown product of cyclic AMP. The cyclic AMP is degraded to adenosine, but there's a little bit of controversy whether adenosine is a morphogen or not. Then volatile ammonia. Ammonia is a gas, and volatile ammonia is also acting as a morphogen, and differentiation-inducing factor. It's a chlorinated hexaphenone that is also acting as a morphogen. Cyclic AMP and ammonia, they are known to promote the pre-spore forming pathway. Adenosine, it's still controversial, whereas differentiation-inducing factor, it promotes the stalk cell forming pathway, the stalk here. So what the structure that you're seeing here is approximately 1 millimeter or anywhere between 1 millimeter to 4 millimeters or so. So my research right now, what I'm going to talk to you today, where is this ammonium coming from? The role of ammonia has long been recognized in Dictyostelium. So this is a picture that I took from the paper published several decades ago. So there's a gradient of ammonia that was known to exist, the gradient of ammonia, whatever. So where is the source of ammonia for all this Dictyostelium? Because ammonia is also toxic in certain, but ammonia is produced in millimolar quantities in Dictyostelium. And where is this ammonia coming from? So when they quantified proteins, overall level of proteins over a period of time, the proteins levels were dropping over a period of time beyond a period of starvation. So subsequent to starvation, if you quantify the total proteins, the protein levels were coming down, and then they assumed that the degradation of proteins is responsible for the origin of ammonia. And this was for a very long time, this was the view. And now we have a different view also. Maybe both of us are correct, we do not know. So adenosine, adenosine is derived from cyclic AMP. Adenosine is derived from cyclic AMP, and there is an enzyme called ADGF, adenosine deaminase related growth factor. Adenosine deaminases are enzymes which acts on adenosine, releases ammonia as a byproduct. So this ammonia is responsible for the further morphogenesis, or is acting as a morphogen. This adenosine, this is called adenosine deaminase. This is adenosine deaminase related growth factor. This adenosine deaminase related growth factor is extracellular. It is made outside the cells. So it is, the enzyme is thrown out of the cell and it is acting on the extracellular adenosine. The ammonia is formed extracellularly and drives tip formation or drives organizer formation. Then we wanted to find out, so when we started this project, there are many deaminases. Deaminases means which acts on the amino group and then releases ammonia. So which deaminase to work on? So one of the deaminase that caught our attention was adenosine deaminase related growth factor, just because of the fact it is extracellular. And in Dictyostelium genome, there are 4 adenosine deaminases annotated. And this is the adenosine deaminase that we are working on. There are 2 independent insertions here. This is a blastocidin resistance cassette, which is integrated here. There is one insertion. There are 3 independent insertions here in this adenosine related growth factor gene, and all of them give the same phenotype. So then we went on to, then we pursued with this mutant. We obtained this mutant from the stock center. It was not created by us. So now we'll run through another movie here. Do you see this? This is aggregative. First of all, what you see here, this is a mutant. The mutant is slightly larger than the wild type. This is the wild type, and this becomes a large. So it spins on its own axis like Vishnu Chakra, and it keeps on spinning and then spinning and spinning for a very long time. Then it gets arrested. And a fraction of them go on to form fruiting bodies after 36 hours. After a very long time, a fraction of them goes on to form the fruiting body. So I keep on emphasizing this. So what is happening here in this mutant is you have a block here. If you have a block here in this pathway, then you expect adenosine levels to go up, ammonia levels to drop. So is it because of high adenosine levels this is happening, or is it because of the absence of ammonia this is happening? So now these are series of pictures. What you see on the top panel here, this is the developmental profile. This is the wild type. This is called the mound. It is going to form at 16th hour. It is already showing signs of fruiting body formation. So by 24 hours, it is from fruiting bodies. That is a terminally differentiated state. Whereas this is the mutant. The mutant is there as a mound, continues to stay as a mound for a very long time. So they are not dying. So a fraction of them, they form fruiting bodies after a long period of time. You can mimic this phenotype by having a ADA inhibitor. This is the inhibitor which is very specific for the ADA enzyme. If you add this inhibitor, then you essentially mimic the same phenotype. So that is the first line of evidence to show that the gene that we are working on is indeed the correct one. Then the second line of evidence is this one. So this is the developmental profile. So it's not moving. Yeah. So we have to confirm that the mutant that we are working on is not showing any expression. That is a confirmatory test. That is, they are working on the right gene. The gene that is in the wild type, it is the peak in expression comes at 16th hour. It is roughly corresponds to the tip formation, the organizer formation that happens at this stage. And we wanted to see whether the other isoforms of ADA, these are 3 other isoforms of adenosine deaminases, are they trying to compensate the loss of this mutant? Or they trying to, because in the absence of ADGF, these adenosine deaminases can try to compensate the loss by increased expression. So when we checked it, it's not happening either. By RT-PCR, we checked it, it's not happening. Then, coming back to this, so what is happening here? Here you have a block. If you have a block, this block in this, adenosine levels are going to go up and ammonia levels are going to go down. So we checked the adenosine levels using a commercial kit, and the adenosine levels at 16th hour, it's significantly higher in the mutant compared to wild type. This is total adenosine. This is not extracellular adenosine. This is total adenosine. And the ADA activity also comes down. The ADA is the adenosine deamination. This is an enzymatic activity. It drops drastically. It is not completely abolished because there are 3 other isoforms which are still have a basal activity. Yeah, that may be the reason why you still have a basal activity. Do you see a movie here? Do you see a kind of, once again, this is a cyclic AMP. So cyclic AMP is one of the morphogens identified in Dictyostelium that plays fundamental roles till tip formation, till floating body formation. So we wanted to check whether what is happening to the cyclic AMP levels here. The cyclic AMP levels are low here in the mutant compared to wild type. We measured at different time points, and 2 different time points at least the cyclic AMP levels drop drastically. Here we can visualize the cyclic AMP wave pattern also by dark field optics. It's an indirect way to visualize cyclic AMP. For example, if you visualize under dark field optics, what is happening is cells which are elongated and cells which are round, they are going to scatter the light in different ways. So one you are going to see the light band and the dark band. So based on this, over a period of time, if you take a movie, then you will be able to see such man. Here you can see a spiral wave, whereas in the mutant it is a circular wave throughout. So initially they start as a circular wave, then there's a symmetry breaking happens, then it forms a spiral wave. That doesn't happen in the mutant at all. So ADG of mutants also forms large size aggregates. Why Dictyostelium also people use this as a model organism to know how tissue size is regulated. An aggregate or a mound or a slug is of certain size. If there are genes going impaired in this pathway, the size regulation is something goes wrong in that process, then the size becomes bigger. So ADG of mutants form large size aggregates. As you can see, the size, when you have a large size aggregates, obviously the number of aggregates comes down, and it is because of we found this was not the main focus of our work. We found what is the reason for increased aggregate size. It is because of increased cell addition, and there are genes impaired in involved in genes involved in tissue size regulation. Their expression, these are some of the genes involved in tissue size regulation. Their expression is altered, as a consequence of which the size goes wrong, and then you have a large sized aggregate. Then coming back to the mutant, what is happening here? What is you wanted to check further? What is happening to the relay, cyclic AMP relay? There are various genes involved in cyclic AMP relay. So the first cell that undergoes starvation makes cyclic AMP. The second cell perceives it. Then it relays the cyclic AMP signal to the third amoeba. Thus, all the amoeba come together. So there are various genes involved in this pathway, various proteins involved in this pathway. So we looked at the expression of various genes. This is called the phosphodiesterase. This is also a phosphodiesterase. This is also a phosphodiesterase. These 3 phosphodiesterases, one of them is membrane bound, one of them is intracellular, one of them is extracellular. These are phosphodiesterase. This is 5' nucleotidase, which is involved in adenosine formation. That the expression level, so this generates this. If this gene is very highly active, you get excess adenosine, and that is what is happening. CAR A is involved in cyclic AMP receptor. GBFA is a transcription factor involved in tip formation. So we looked at various genes, their expression profile over different time points. And coming back to the wild type. So now is the wild type, that's the mutant which you can see. Now, can we take the supernatant? The supernatant, we call it as conditioned media. Can we take the supernatant of this wild type and see if we can add the supernatant to the supernatant, the AX4? For example, this is AX4 supernatant on AX4. This is the control. The same control goes here. Whereas if you take the wild type supernatant and then add it onto the mutant, then it gets rescued. Similarly, if you take the conditioned media or the supernatant of the mutant and add it on to the wild type, you have the same developmental block. So something is secreted, and something is secreted as a consequence of which you have this problem. So if something is secreted and if the secretion is not proper, then you'll have the problem like this. You can also mix wild type. Here it's a medium supernatant. You can also mix the wild type with the mutant, physically mix them. And if you mix them in a 20 to 80% ratio, you have partial rescue, whereas 50 to 50%, you have a complete rescue. Now, is it because of adenosine deaminase? So we took the mutant here. Here is a mutant directly on top of these on top of this structure. We add the human ADA enzyme. This is a commercially procured enzyme. So this enzyme directly we add it onto the top, then it is a rescue. So if you have more you add it, it's complete rescue. This is also getting rescued with caffeine. What does caffeine do? Caffeine is an antagonist of adenosine. Antagonist of that is also getting rescued. And this is a 8-bromocyclic AMP. 8-bromocyclic AMP is a stable form of cyclic AMP which gets into the cell and stays for a longer period of time. That is also getting rescued. So this is already suggests you that cyclic AMP is the main player. And cyclic di-GMP also rescued. The reason why I emphasize cyclic di-GMP is because of cyclic di-GMP is a prokaryotic signaling molecule. And that is also used by eukaryotes. And that induces the organizer formation. That induces the pre-stock formation. That was published by someone else many years ago. See, when we check with cyclic di-GMP, that also induces. But as the concentration goes up, you can see some multiple tips. So instead of one tip, you get multiple tips also. If you go back to this reaction, adenosine, it forms ammonia. So if you have a block in this pathway, then ammonia is not there. So now we ask the simple question, can it rescue the phenotype? So what we did was, here you have a plate like this where you have the mutant. On the top lid, where you can have here you can generate ammonia artificially. If you generate ammonia artificially, then what happens? There is a rescue which is happening. So in all the concentrations we tested, the ammonia is getting rescued, and the ammonia levels, when we tested, the ammonia levels are also lower in the mutant, but when we add it externally, so the ammonia is not physically, it is there in another lid. So found it is able to rescue the phenotype. Now, this is an interesting aspect. Now, wild type without physically touching the mutant. So wild type, what is happening? Wild type should produce ammonia, whereas the mutant is not producing ammonia. So what we did was, we took a compartmentalized petri dish. On one side we have the mutant, on the other side we have the AX4, which is the wild type. AX4 is the wild type. Now we asked a simple question. If it is genuine, if ammonia is indeed responsible for the rescue, can it rescue the phenotype here? Because here you have a compartmentalized dish. So it is able to rescue the phenotype here. So without physically touching. So this is happening. And what is interesting here, now we asked another question. I'm not sure if I have the slide, but I'll come back to this later. We also added bacteria here in the one side of the petri dish, on the other side the mutant. Bacteria, if you have bacteria on a non-nutrient agar, the bacteria is going to die. When they die, it is going to generate ammonia. The dead cells generate ammonia. When they generate ammonia, it is going to rescue the phenotype. So bacteria also rescues, but after a certain time. Wild type rescues in a period of 4 to 5 hours. And this is the idea of conditioned media. So if you can take the conditioned media, that is the supernatant, you can add this ADA enzyme and see if ask the simple question whether it rescues. There also it is rescued. Finally, the gene, the overexpression. If you take the mutant and then compensate this with the overexpression construct, you take the wild type copy of the ADGF and then put it here, then that is also able to rescue the phenotype. So multiple lines of evidence. Yeah, please.
Can you once again explain the setup in that like how
this is a partition petri dish. That means there is a physical compartment running through. That is what you see here. On the one side you have the mutant. On the right side, on the left, for me it's right side. Yeah, from the right side it's a mutant, left side it is a wild type. Now we ask a question, is if ammonia is produced by the wild type, can it rescue the ADGF mutant? And the rescue happens.
So ammonia is diffusing through the partition.
So it's not completely sealed.
Okay.
It's like the porous structure here, what you see on the top. The ammonia goes low.
So apart from ammonia, other morphogens can also diffuse
No, through the air.
through this partition. Oh, okay. Ammonia is volatile. Yeah.
Ammonia is volatile. That's why it's able to rescue. The others are not.
Okay. Thank you.
And I'm not, the temperature is at 22 degrees Celsius. So
Is it easy to see if the mutant has been rescued?
yes, it is rescued because I see the tips here, several tips here.
I see.
And also the size slightly goes down, because the site is here. It's all, you can see these are all like pebble stones, The pebble stone, you no longer see that kind of pebble stones here. Now, we have not proved, so far we have not proved. So even though I took the wild type and then have the mutant on the side, we have not proved so far that adenosine deamination, that's a chemical adenosine. From the adenosine, there's the amino group is cleaved off and then it is responsible for ammonia formation. So similar to the last experiment, here we took the mutant. On the left side, we have the buffer. So the buffer, it's the ice-cold buffer. To this, we add the ADA enzyme. ADA enzyme plus adenosine. So on one side, you have the adenosine plus buffer. And the right-hand side, you have the moment you add the buffer, after 4 hours, you get, so this is getting rescued. So all of them are rescued now after a few hours. So this convincingly demonstrates that adenosine deamination alone drives the rescue. So downstream of it is cyclic AMP. So now we took several mutants which is having this kind of phenotype, like pebblestone kind of phenotype, and then took these mutants because they show a very similar to. Why do we take this mutant? To know what is the pathway by which this gene works, the ADGF works, pathway by which mound formation, tip mound formation or organizer formation takes place. So we took several mutants of this, mutants which looks like this, and then directly added ADA enzyme on top of it. When we added, then we found that this is a histidine kinase. Histidine kinase this is a mutant impaired in histidine kinase. If you have a block in this, then it is manifesting in mound arrest. So if you treat them with the ADA enzyme, then you are able to see a rescue. The more you add it, the more then you can see a multiple tip phenotype. So this is a pathway by which we are able to find out. We tested several mutants, of which only we found DHKD, and these are other mutants. When we added this ADA enzyme, there was no rescue. Similarly, what we did was we took mutants with multiple tip phenotype and added. Then we did the reverse. We tried to block the ADA activity by adding the chemical inhibitor. When we did this, there is no rescue either. So of course, we have not comprehensively looked at all the mutants available. We took a range of whatever that is, dozen or half a dozen mutants or so. Now, DCF, so is it affecting the pre-stock, pre-spore pattern here? Pattern, that what? Already this is the pattern, the pre-stock, pre-spore pattern is, so this is the slug. The front end of the slug is going to die and form the stalk. Here you have a molecular marker. This marker is called the extracellular matrix, and that is tagged to GFP. And when we add the drug, when we chemically block this ADA with a drug called DCF, then you see a slightly enhanced expression, and you also see a some pattern of expression at the back of the slug. Conversely, you can take the pre-spore, that is the other end of the, so this is pre-stock, this is pre-spore in the wild types. So what I see, this is the front end and this is the back. What you see here is the front is the pre-stock. What you see on the back is the pre-spore. So now when we did this for pre-spore, what is happening here? This is the wild type. After adding the drug, there is no change in the pattern. But when you take the neutral red marker, this is the front side, and this is the back side. But in all of them, there is a slight, and in fraction of the slugs, there is a small bit of increased expression of pre-stock cells on the back of the slug. And you are able to see when the pre-stock marker expression, the ECMA marker, which you see here, that is the spatial pattern. There is no change. But however, There's a slight increase, there's a reasonably increase in pre-stock expression in both the cases. Probably these cells may account for the increased expression here. And pre-spore expression is down-regulated in both cases. Now, ADGF cells have high levels of adenosine. The view is that high levels of adenosine at the slug front is suppressing one more tip along the axis. So we took cells with high levels of adenosine, that is the mutant. This is the wild type on the top panel. We are labeling with a cell tracker dye. This dye completely stains the slug with red color, which you can see here. So this is the wild type. This is the mutant forming a large colored slug, large size slug. When we the ADGF cells, which is carrying high levels of adenosine, when they are mixed with the 80% of the wild type cells, the cells carrying high levels of adenosine goes to the slug front. Conversely, when you do the reverse experiment, you can see scattered staining here. Now you can ask me why the staining is scattered here. Because we are doing with 20% cells here, not 80%. If you add 80% labeled cells, then the slug will be glowing. Then it will be difficult to visualize the fluorescence. Now, how is this gene regulated? The gene has to be regulated by some means or other. So we took the wild type cells. The wild type cells, this is wild type cells. They are treated independently with the adenosine or ammonia. This is the substrate. This is the product. Then we looked for the expression of various genes involved in this, various genes that is of interest. One is the adenosine deaminase, the same gene that we are working on. This is in wild type. Then this is the gene that is responsible for cyclic AMP synthesis. And this is the gene that is responsible for cyclic AMP degradation. What is interesting here, which if you can draw your attention now, here is you see that from AX4, as the concentration of adenosine goes up, there is a gradual decrease in phosphodiesterase expression with the increasing levels of adenosine. Whereas with the increasing levels of ammonia, the levels go up, completely contrasting expression. And ACA expression, which is responsible for cyclic AMP synthesis, depending on the dose that you can see, as well as the ADGF, depending on the dose that you see, there is differential pattern of expression. So now we asked if adenosine is involved in suppressing tip development, if you add adenosine to wild type, will it suppress tip development? Adenosine is responsible for, that is the hypothesis we start. However, when we added adenosine, it is not doing substantial changes to the tip formation, no change here. Adenosine after 10 hours, no change in the wild type. The derivative after the, if ADGF really works, inosine is also found. Inosine also has no effect. Related purines we also checked with guanosine and deoxyadenosine. They also don't have any, they also don't exert any effect at all. So mutations in other genes encoding for intracellular ADA. Intracellular, this is not extracellular, this is intracellular ADA. That is also forming tipless mounts, but this is still the results are not confirmatory because this is still, we assume that this is happening. We have to do additional tests to confirm that this is the case. We obtained further, we obtained several classes of mutants. All of them, these are knockouts. These are knockouts involved in other deaminases. So this should also generate ammonia. And if you have a knockout here, they also manifest in the mount arrest, but the mount arrest phenotype is not as pronounced as in ADGF, the extracellular. They show a partial mount arrest. We took the AlphaFold, which is the online software, and we modeled the structure. It is very similar to the human, this is called cat-eyed syndrome. The human ADA2 is very similar to the ADGF of Dictyostelium. So Dictyostelium can also be used as a model for cat-eyed syndrome, studying cat-eyed syndrome. This is the Dictyostelium ADGF. This is the human ADGF. This is the overlapped structure of, the structures are aligning well. And the structures are aligning well. Then we wanted to check what is ADGA, human ADA interacting with. So human ADA is known to interact with dipeptyl peptidase. Human ADA is known to interact. So the equivalent of dipeptyl peptidase in Dictyostelium is called APRA. We obtained the mutant, and when we abolished the function of adenosine deaminase, We found no phenotype here. So maybe this is the phenotype may be apparent in the growth stage, but not in the developmental stage. So this is the dipeptyl peptidase. It is there is a strong interaction of dipeptyl peptidase. That is dipeptyl peptidase is the APRA with the ADGF. And this is the DHKD, which I said which is involved in the histidine kinase, which is involved in the pathway. So now, when we looked at the online database of human gastrula or mouse gastrula, during gastrulation, the organism development happens. During early gastrulation, organism. So the adenosine deaminase expression is stronger in both these embryonic stages, and but when we looked at literature, not much is known about adenosine or ammonia formation during early gastrulation. So the very simplified model of how this works is like this. Cyclic AMP is the first messenger as well as the second messenger. Cyclic AMP is thrown out of the cell. It is secreted. This cyclic AMP is the adenosine is derived from cyclic AMP. From adenosine, ammonia is formed. So ammonia may be coming from protein catabolism, enzymatic reaction, adenosine deamination, several reactions. Even if you have a block in one, it is manifesting in the mound arrest. So this is the mound arrest means what? There is no organism formation. There is no tip formation. So the low adenosine and high ammonia levels that is responsible for genes that induces adenyl cyclase expression that makes more cyclic AMP. The cyclic AMP relay is proper. Then cyclic AMP wave is happening, as a consequence of which you have a proper tip development or organism development. So I thank my guru for introducing me to this great organism Dictyostelium, and I also thank Professor Nanjundaya for introducing me to another great professor, Professor John Bonner. And this work was entirely done by Pavni Hati. And you see, these guys are judges, and they are all biased like me. And this is Dictyostelium, which is walking here. And all of them give a score of 10 out of 10. And if you have time, we can go on to the second part.
I have a small question. So I'm still not completely clear about. Can you just explain the role of ammonia in the formation of the tip in the tip development?
Ammonia induces cyclic AMP.
Okay.
And cyclic AMP triggers cell movement. So cell movement is necessary. And if there's a block in ammonia, whatever cyclic AMP is, not enough cyclic AMP is being made. And cell movement, collective cell migration, collective cell movement, this is also happening during gastrulation. And that collective movement is blocked, as a consequence of which tip is not formed.
Okay.
So ammonia comes first, then cyclic AMP. Then cyclic AMP triggers cell movement. That is what is happening.
Okay, so for this tip to form, is some sort of a differential concentration of ammonia needed over the cells, or is it?
That part I don't know. We do not know.
Okay.
Differential but what we think is ammonia is being made throughout. And there is something called slime sheet. If this is a slug, there is something covering on top of it, exactly like my hand. The front end is exposed, so there is lot of volatile ammonia that is getting released. Whereas the back, the diffusion is limited.
Right.
So that is a gradient. But there is what we do not know is what is the role of ammonia in the surrounding environments. Because even bacteria, the nearby bacteria, dying bacteria, they can also rescue this phenotype.
Thank you.
So when we started with this, initially when we started, I started with this model organism called Polysquandalium. This is a fantastic model to work with, but it's very notorious because this forms symmetry breaking. Symmetry breaking, if you want to investigate, for example, here you see a round structure here. The round multicellular structure within a span of 40 minutes, it breaks its symmetry and forms a spheroid. From spheroidal symmetry, like a spokes on a wheel, it forms a structure like this, so from globular to spherical symmetry. But this organism is very notorious for transformation. So if we cannot do transformation and do gene manipulation, then it's very difficult. But this model organism is very interesting because it is not making cyclic AMP as a chemoattractant. Instead, it is making a modified dipeptide called glutamic acid and ornithine as a signaling molecule. So initially when we started, we looked at the role of caffeine in this problem. That led me to working on adenosine deaminase. This is a slug. If you transplant this slug and put it onto a plate containing caffeine, then what you have is multiple heads that are getting formed, multiple tips that are formed. Normally you have only one tip, that is at the front end. But when you put it here, then you get multiple tips. So this is a very nice problem to look at lateral inhibition. So high levels of adenosine, caffeine is an antagonist of adenosine. High levels of adenosine and the slug front is not allowing one more tip that is going to be formed along the slug axis. When the adenosine activity is blocked, then you have multiple tips that are formed. This is the typical form of lateral inhibition that you can see here. Sometimes you really wish you could do this. So I'll skip this for the sake of time. So this is a typical problem to look at. This is a fantastic problem from globular to spheroidal symmetry. Nothing happened. It's didn't work for us. So coming to Arabidopsis, this is a model plant. I'm going to. So this is the X-ray image of a nice Arabidopsis flower. What we do is there are 4 parts of a flower. This is the sepal petal. This is the male part, and this is the female part. Upon fertilization, you get a lot of seeds here. And so here we try to estimate spontaneous somatic mutation rates in Arabidopsis. Spontaneous somatic mutation rates, even with sophisticated sequencing, it's not possible to estimate these classes of mutations because these events are very rare, happen somewhere in the structure, somewhere in the plant. So how we detect this somatic mutation is very simple. So you transform the plant with a marker. This marker, when it is expressed, the whole plant will be blue in color. But we deliberately make a mutation. In this, if you deliberately make a mutation, you can make a mutation precisely and introduce this back into the plant. Now, if this goes to a reversion process, that is, a mutated gene goes to wild type, then you see a blue spot here. So, based on the number of blue spots to the total number of seedlings that you screen, then you can estimate a rough estimate of what are the mutation rates. So, here we looked at these kind of vectors. For example, here this gene is mutated in such a way that it carries point mutation. So, you introduce a stop codon there. If the stop codon is there, then the message is going to be truncated. The truncated message, it does not make any sense. So, as a consequence of which you will not see. Suddenly, if it goes through a reversion process, then you will see upon staining, you will see a spot like this. Similarly, here you have a microsatellite. The same, for example, here you have stretches of G's introduced. And for example, if you have a stretch of 8 G's introduced here, either by addition of 1 more G or deletion of 2 more G's, the frame can be restored. So that way the frame can be restored. This is a microsatellite introduced. So once again, the reversion. Here is a transposon. In between a promoter, global promoter, followed by a reporter gene, there is a transposon sitting here. If the transposon jumps out, then the gus is going to be formed. Then you are going to see a blue color. And the last one is how we estimate homologous recombination. Here you are the gene is called the gus. If you can look at my hand, it is truncated and then introduced like this. If it goes through a process called homologous recombination, then you have the reversion, then the reversion can be visualized here. That is one problem that we are looking at. What we are looking at is how is hybridization, because Arabidopsis comes from a different source, how is hybridization influencing the spontaneous somatic mutation rates? So we took Arabidopsis from different parts of the globe. All of them you can collect, you can order it from the stock center. We made certain number of hybrids, generated stocks, analyzed the mutation, and also this is published now. So hybridization, we asked whether hybridization triggers one class of mutations over the others. But in all cases, we found out that the parental sex makes a huge influence on the mutation rates. Then the second so we are looking at 3 problems in plants. One is how hybridization is influencing the mutation rates, how parental lineage reproduction is influencing the mutation rates, how seed age is influencing the mutation rates. Because seeds stored for a very long stretches of time, and if you store it for a very long time, the reactive oxygen species levels also are it is published. Their levels are also known to go up. So do they affect the spontaneous mutation rate? The second one is how we estimate meiotic recombination rates. The assay is very simple. If you can focus your attention only on this set of chromosomes, these are called meiotic recombination detector line. So this is a homologous chromosome. In this chromosome, you have a RFP, red fluorescent protein, that is driven by a seed-specific promoter. So that, what do you mean by that? If this RFP is expressed, you are going to visualize this in the seeds. And this is GFP, this is green fluorescent protein. This green fluorescent protein, you are going to visualize the seeds. Suppose if there is a crossover, there is a single crossover here, only one crossover. Then you are going to they are going to get separated like this, which you can visualize here. Then, for example, this green here, that you visualize the same set of seeds under RFP filter and a GFP filter. For example, if you look at green, this green is not reflected here. This is R. That R is not reflected in this portion here. So based on this, based on the fraction of red only and green only, you can estimate the meiotic recombination. But this line is not perfect. This has got pitfalls because the length, if there is a double recombination happening between these two markers, then it's very difficult to visualize. There is no way you can distinguish. So we are having markers that are very close by to each other and working on the effect of seed age as well as the role of plant hormones in meiotic recombinations. We found that strigolactone is an hormone that plays a significant role in meiotic recombination. Stop. So I'll stop here. Thank you.
I have two questions. One is, in that experiment where you added bacteria to slime mold plates,
Bacteria are independent. They are not physically touching.
beg your pardon?
They are not physically touching the slime molds.
Okay, so you answered my question. I was wondering whether they reverted and started feeding.
No. They compartmentally, one compartment you have bacteria, but the bacteria takes time to die. So the recovery...
I get it. Yeah. Thanks. The other question was, quantitatively, how much of the ammonia produced is from cyclic AMP and how much via protein degradation?
We don't have an answer for this. Good question, but we don't have any answer at all.
Further questions? Yeah. Maitre.
Is there anything qualitatively different when, do ammonia and cyclic AMP change? I'm just wondering if the phenotypes of your adenosine deaminase and not having cyclic AMP have identical phenotypes.
Cyclic AMP, what you're saying is if there's a block in cyclic AMP production, will it give a similar phenotype? The answer is no, because cyclic AMP is involved early in development. So that means very early in development, even chemotaxis will not take place.
But so is morphogens, cyclic AMP and ammonia,
they interact because more ammonia is being made, it triggers adenyl cyclase expression, which is responsible for cyclic AMP production. Yeah.
So we saw that adenosine and ammonia both are kind of affecting how this will happen, like the tip formation will happen. So is it some kind of the ratio of adenosine and ammonia that is guiding?
That is what initially we thought, but that doesn't seem to be the case, because when you add ammonia, in spite of high levels of adenosine sitting in the plate, ammonia is able to rescue. Even at smaller concentration, even bacteria is able to rescue. So adenosine, the role is not very clear. It's very controversial. In literature also sometimes they say yes, sometimes there is some other group that says no. Adenosine, it's not. Whereas ammonia, we can categorically say. The ratio, I do not know. Is it the ratio of adenosine to ammonia?
Adenosine independently cannot do.
That is what, if you add adenosine to wild type, okay, then it should induce the Mounder's phenotype. That does not happen.
Okay.
Could it be because of the wavelength of the waves? I mean, basically you have a much larger spacing between successive waves in mutants. Have you tried measuring it? No. Because if the refractory period, excitable medium. Yeah, so basically they have to, so for example, in excitable media models, by altering the refractory period, you can actually make spiral breaking, but breaking into spiral is very hard in Fujinagumo and so on. Yeah. So I'm saying that in Fujinagumo, for example, breaking a circular wave into spiral becomes very hard if you make the refractory period, alter the refractory period. So maybe something like that might be happening here. So you could just try measuring the spacing between successive waves and see if that changes. Yeah. Any other questions? So I'm just wondering, suppose, once the Dicty have started sending out the cyclic AMP, if at that point you suddenly add again a lot of bacteria, can it stop the cyclic AMP signal and revert back to feeding the bacteria, or is it like a irreversible thing? Once they decide
Not the slug stage. The slug stage you cannot do it, but the aggregation stage I do not know.
I see.
Slug stage I think they ignore.
All right.